Alright, today we're going to be talking about the genetic code, and we've been learning about the central dogma of biology, which is that DNA makes RNA, which makes proteins. We also know that DNA can replicate itself. We've been learning about that process, and we know that the DNA is the template that makes the mRNA. The mRNA is the template that makes the protein. So today we're just going to be talking about the genetic code. This is the code that tells the cell what kind of protein to make from the segment of DNA. So let's start off with a few terms that you may not have heard before. The first of these terms is codon. So the word codon means you have three nucleotides that are next to each other in a sequence of DNA or RNA that encode for one nucleotide, like tryptophan or leucine or valine or something. Amino acid, not nucleotide. So three nucleotides make one amino acid, and that's a codon. That's what that word means. An open reading frame, or ORF, is the section of the DNA or the mRNA that is actually going to make the resulting protein. That's called an open reading frame. As you can see, we have a start and a stop, which encodes this area of the mRNA called the open reading frame, and that's what's going to make a protein. And then there's another word that I'm going to be using, degeneracy. And this word doesn't actually have the same meaning in biology in terms of the genetic code that we would normally think of the word degeneracy or degenerate meaning. So in our case, it actually means that there's the ability in the genetic code for one amino acid to be encoded for by multiple codons. And we will talk about all these things in a moment. So first I'm just going to give an overview of what we'll be talking about today. So we're going to learn about the genetic code, and we'll learn that the genetic code is composed of nucleotide triplets, or codons. It's non-overlapping, it's comma-free, it's degenerate, ordered, regulated by start and stop codons, and it is universal, or nearly universal, genetic code. So let's talk about these nucleotide triplets. So I've given the class a handout, which is also represented here on this slide, which just shows us the composition of the nucleotide triplets. To read this chart, one starts in the middle with a G, a U, a C, or an A. And remember, we're in mRNA, which has a uracil instead of a thymidine, like DNA has the thymidine, T, instead of the uracil U. So G, U, C, or A. And then you can have a G, A, C, or U, and then again a G, A, C, or a U. So to read this, if you're looking at a codon, which is, remember, three nucleotides, you start with the first nucleotide, let's say it's a C, then you look at the second nucleotide, let's say that's a C, 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 and maybe the third one's a U, C, C, U. So you would find C, C, U, and you would see proline. Aha! C, C, U means proline. So that's how to look at this chart. So who came up with the notion that the codon is three nucleotides? How did scientists figure this out? So first there was some logic involved. So we know that there are 20 amino acids total in the cell. And we know that there are only 4 letters in DNA or RNA, which are A, T, C, and G in DNA, and in RNA it's A, U, G, C, right? So there's 4 letters and 20 amino acids. So we have to be able to make 20 different amino acids from just those 4 letters. So we know that if we only have 2 bases per codon, which is, could be 2 bases makes 1 amino acid, that would be 4 to the second. And that's only 16 amino acids. So we can automatically rule out that number because that's not enough to even make the number of amino acids in a living cell. So it has to be more than 2. So the next number, obviously after 2, is 3. And we can see that 4 to the third is actually 64, which is way more amino acids than the cell actually has. But it has to be at least 3 in order to make 20. So scientists knew that while they were trying to figure out exactly what was the genetic code. So one major experiment that helped us figure this out was done by Francis Crick, the same guy who, with Watson, discovered the structure of DNA. And Crick and his colleagues in 1961 did a very clever experiment using a bacteriophage, T4, which could infect, in its wild type form, 2 different types of E. coli bacteria, strain B and strain K12. However, when Crick and colleagues mutated this bacteria, or the bacteriophage, it could only infect strain B. But they realized that if they continued to mutate this phage, and the way that they would mutate the phage was to either add or delete a nucleotide in the mRNA sequence. If they mutated this once, then it became a mutant phage and could only infect one type of E. coli. If they did it twice, it could still only infect one type of E. coli. But if they did it 3 times, some of those phage could actually go back and infect both types of E. coli. So they were able to realize that if they added or deleted 3 base pairs in the phage, somehow they reverted the phage back to its wild type phenotype and allowed it to infect both bacterial strains. So in this way they were able to logically deduce that there must be 3 nucleotides per codon. So that was one of the first experiments that scientists did to try and figure this out. So 3 base pairs equals 1 amino acid. Another clever experiment was done also in 1961 by J. Heinrich Mathai and Marshall Nirenberg. And they were able to take strings of nucleotides, for example, U, and put this into a cell containing radio-labeled amino acids, and figure out which amino acid got incorporated into the protein by using U. And so they figured out that this makes FFFFFF, which is polyphenylalanine. Okay? So they were able to say that a string of U's made phenylalanine. And they were also able to go and do this for all the other combinations of codons that they could come up with and figure out basically the genetic code. And this was how they figured it out. So this was pretty clever stuff for the beginning of the 1960s. So those experiments and others gave us what we know as the genetic code, which is these 3 nucleotides make 1 amino acid, and they were able to discover all these codons and which amino acid they coded for. All right. We also know that the genetic code is non-overlapping and comma-free, which means there's no pausing in the middle, and the code is read in sets of 3, which do not overlap, as I've indicated here. So we have 3 AUG, then the next 3 CGG, then the next 3 AUG, then the next 3 AAC, and so on. The code does not ever go AUG, UGC, GCG. That doesn't happen. It's always sets of 3. All right. The genetic code is degenerate and ordered. What do we mean by that? Well, as I've shown here, for some amino acids, they have more than 1 codon. And this makes sense because, as you remember earlier, we said that 4 to the third was 64. There's only 20 amino acids, which means that there's 64 possible codons making 20 amino acids. There has to be some repetitiveness here. And that's exactly what we see. So for leucine, you'll notice that it's really the last nucleotide, the third in the codon, that's really different. Okay? So it's UUA, UUG. UU is the same. Then CUU, CUC, CUA, CUG. CU, something else. And the same is true for serine. Now, there's also amino acids that don't have 6 codons. They maybe have 2 or 1 or 4. So for example, histidine only has 2, but you'll notice that the 2 that are different are both pyrimidines. U and C are both pyrimidines. And for glutamine, A and G are both purines. So this means that even though the cell makes a mistake every now and then in DNA replication, it doesn't have to be a bad thing. If it makes a mistake in the 3rd part of this codon, it's still going to be a leucine. No big deal. All right? So then that helps the code be degenerate, right? That's what that means. Now you notice for some of these codons, there's only 1 amino acid per codon, or 1 codon per amino acid. So methionine, AUG, is only AUG, and that's it. Nothing else makes methionine. All right. Degenerate and ordered. So now ordered, what do we mean by that? Amino acids with similar properties have similar codons. Okay, so for example, leucine, isoleucine, and valine are all hydrophobic amino acids, and they look very similar in their 3-dimensional structures. And if you'll notice, they have very similar codons: CUU, AUU, GUU. So if the DNA replication machinery makes a mistake in the first codon, or the first nucleotide of the codon, and puts in maybe an A instead of a C, it's still not a horrible thing for the cell, because the cell can still put in an isoleucine in the protein instead of a leucine. And in the big picture of a whole protein, that's not a big difference. That's not a big change. So the protein will probably still be functional and go on and do its normal thing. And so this is how the code can be degenerate and ordered. So things can be replaced, mistakes can be made, but still, usually, things turn out okay. The genetic code contains start and stop codons. All right. So for the start codon, there's only 1, AUG, methionine. You should know this. This is a very important codon. If you are going along, if you have a stretch of amino or a nucleotide sequence and you see AUG, you know, aha, this is a start. Okay? Not every AUG is a start, because there's methionines throughout the mRNA, but the first time you get to an AUG in an mRNA molecule, that's the start. Okay? And there are 3 stop codons: UAG, UAA, and UGA. My high school bio teacher gave me a great way to remember these codons. If you pretend you're a caveman and you say UAG, or UA, or UGA, you're a stop codon, all right? And the area between these start and stop codons is the open reading frame. So here's our start, AUG. Here's our coding sequence, or open reading frame. And here's the end, UAG, right? Stop. That's the end. We're not going to make any more amino acid incorporations. Okay. And finally, the genetic code is nearly universal. So all organisms, me, you, orangutans, amoebas, monkeys, fish, birds, we all, E. coli, we all have the same codons for the same amino acids. So AUG in a human, and AUG in a bacteria, methionine. Okay? There's an exception, an important exception to this rule, and that's that in mitochondria, UGA is tryptophan, AUA is methionine, and the AGA and AGG means stop. So that's the difference. All right, so let's review what we've done here. We've shown that Crick and colleagues have shown evidence for a triplet codon. And in the same year, Nirenberg and Mathai demonstrated that UUU was phenylalanine, and they went on to make predictions about the entire set of codons, what they were. We've learned that the genetic code is non-overlapping, it's comma-free, degenerate, ordered, and universal, and the genetic code contains start and stop codons. All right.